John Grabowski, National Estuarine Research Reserve Graduate Fellow, and Meg Dolan, Research Technician, are using remote areas of the Rachel Carson Reserve to study oysters, one of North Carolina's most important seafood harvests.
One of the motivating factors, some of the reasons I've actually been able to get some funding to do this research, is that oyster reef habitat, there's an oyster reef fishery. Simultaneously, we've recognized that the habitat has other values, and we're doing the research to explore that. We've looked at whether or not it enhances finfish and crab and shrimp production, all of which are commercially and recreationally harvested. We're using in our sampling the same materials that the commercial and recreational fishermen utilize, gill nets, the traps, and some of the rod and reel or hook and line, in order to quantify and compare what kind of fish and crab and shrimp catches we're getting on reef habitat as compared to control areas. Therefore, we can go one step further than just to quantify everything that's on these habitats, but also to attempt to put economic value on the different function of the reef, i.e., what is the value of the oysters produced by a reef, and then what are the values of the commercial and recreational fisheries produce on a reef. One of the interesting comparisons ours make, when you harvest the shellfish, you often destroy the habitat, so you might lose in perpetuity these other benefits, i.e. the enhanced fish and crab populations that are created by restoring this reef habitat. So you might be considering a very temporary benefit to the oyster population versus a long-term benefit to the fin fisheries.
John's research will help us understand if it is possible to restore oyster reefs that have been over-harvested. Meg is looking at different organisms living on the reef and how they affect productivity.
What we're really looking at is how the different organisms affect one another. In our experiment, we looked at 3 different organisms: juvenile oysters, mud crabs, and oyster toadfish. The oyster toadfish, if they prey on the mud crabs, then release the juvenile oysters from predation. That was our idea. So that's what we're looking at. So if you have a big pool, right, an oyster reef, and you throw in an oyster toadfish and 50 mud crabs and then juvenile oysters, will the toadfish actually affect the abundance of the juvenile oysters? And we found that yes, they will. And it's not just by eating the mud crabs, but also it's the behavioral effect. So, if a mud crab is around an oyster toadfish, they'll kind of, get down in the oysters and really just kind of hide.
Good science requires precision measurements and experiments that can be duplicated by other scientists.
What we essentially have done is we recreated this habitat that's 5 meters wide by 3 meters long, so it's 60 square meters. And we've taken a quadrat that's a 1/2 meter by 1/2 meter and set it up so that we've tied in monofilament line. And so we put 10 lines on each side to make 100 intersections, so we can sample 100 gridded quadrat plots. So we'll have 100 point counts. So what I'll do is I'll stick this down and I'll count across there, and at every point I'll look and see if there's a live oyster, a dead oyster, some kind of algae, or some kind of other sessile organism that's attached to the shells. As a scientist, you want to be able to quantitatively say what's there. In short, if we just went out and looked, we'd be very biased in our sampling measures. We wouldn't have a very good feel. We'd have nothing to compare by. Science is based on taking measurements so that you can then do statistics, so that you can then be very confident about what you say.
For John and Meg, field work is only a small part of their research. Many hours will be spent in the lab analyzing their data.
It is beautiful out here, and it's wonderful to get to work here, but you'll find yourself so into your work and so caught up in your sampling method that you won't even notice a lot of what's going on around you, which is kind of sad in a sense. For some of the samples we take where you just dig out the reef to see what lives inside the reef, people spent 8 hours picking just a bucket of shell fragments just to find the organisms, and then you spend another 4 hours perhaps IDing the organisms and to species. So there's definitely a lot of time that goes in just doing the lab stuff, doing the measurements, IDing the organisms, and picking through the samples to pick out all the organisms.
My brothers are all interested in coming down and working with us, and they say, you know, they just envision this huge ship and all the scuba equipment you can imagine, and there just is no glamour. Last summer when we were out hauling all of this oyster shell, literally covered from head to toe in sand and mud, and it was just a mess. So the glamour is definitely someplace in Jacques Cousteau land, not here in the estuary.